So once triglycerides, or triacylglycerols, are in your duodenum, they get packaged into chylomicrons that then travel through your bloodstream until they arrive at a cell. And the cell is going to break these down. The first way it's going to break them down is through an enzyme called lipoprotein lipase. And the mechanism for this is a lot like the mechanism for the pancreatic lipase that we saw earlier, except instead of just separating two of the fatty acids from the glycerol, it's going to separate all three. So after this, but the mechanism for those is essentially the same. And so after the action of this lipoprotein lipase, you end up with the glycerol, that's what I'm removing here on the left, that glycerol being one separated piece, so glycerol, and then three fatty acids. Now notice that in blue, everything I'm writing in, if you add it all up, it adds up to 3 waters, right? H2O. So you're really just adding waters here, but here you have 3 fatty acids. And the mechanism for that, like I said, is essentially the same as the mechanism for the pancreatic lipase that was represented in another video. Okay, now each of these have different destinations. The glycerol and the 3 fatty acids are going to be going to different places. The glycerol, well, let's start with 3 fatty acids. The 3 fatty acids are going to be going to the mitochondria, and they're going to be broken down for energy. So that's something that we're going to explore in a separate video, how that happens, how those get broken down to create energy. In this video, we're going to explore what happens to the glycerol. The glycerol gets carried back to the liver. Ultimately, it's going to be broken down as a carbohydrate, so its final fate will be explored in a separate video. In this video, I want to explore how this glycerol changes from glycerol to something that could enter a carbohydrate metabolism pathway. Okay, now the first thing that needs to be done is the glycerol needs to be phosphorylated. A phosphate needs to be put on it. So here we have our classic source of phosphates, adenosine triphosphate. We've got the 3 phosphates on there, and then the adenosine ring like we've seen before. So that's going to be our source of phosphates, and the enzyme that's going to catalyze this is going to be glycerol kinase. In general, a kinase is an enzyme that puts a phosphate onto something. So these are very common things. Phosphates get transferred around all the time to either make enzymes start working or stop working, or to activate or deactivate other things. All the enzymes that put phosphates onto things are called kinases. Enzymes that take them off are called phosphatases. But here we're putting the phosphate on. So this glycerol kinase is going to activate this hydrogen at the bottom here. And it's going to do that with a proton transfer. So I'm going to draw the bond out a little bit more explicitly. And I will also draw dots in there. Okay, so an aspartate, and it's going to be the 245th amino acid in the chain of this glycerol kinase. That aspartate is going to do a proton transfer. It's going to take the hydrogen off of that OH on glycerol. So those electrons will rip off this slightly positive hydrogen there, and the electrons will snap back onto the oxygen of that glycerol. So this lone pair will now be a bond. And at the end of that bond is going to be this hydrogen from glycerol. These electrons are going to snap back onto this oxygen, and that will now be a nucleophile, a strong nucleophile. And so the effect of this glycerol, this proton transfer reaction, is it makes this oxygen a strong nucleophile. Now that this oxygen of glycerol is a strong nucleophile, it can do an SN2 reaction on one of the phosphoruses in the phosphates of ATP. These phosphoruses are bonded to a lot of oxygens. All the oxygens are pulling the electron density away, and so the phosphoruses are really slightly positive. Each of them are. They're losing a lot of electron density to the oxygens that they're bonded to. So they're vulnerable electrophiles. Notice also that if one of them attacks, the one that leaves, that gets kicked off, would be a good leaving group because the negative charge will be stabilized through resonance. You'll have an allylic lone pair. I'll draw that out in a second. Now, in order for this nucleophile to get close to the phosphates to attack, it has to overcome some of this negative charge down here on these oxygens. And that's where the enzyme positions a magnesium ion close to those. So this glycerol kinase positions that magnesium ion close to these two end oxygens. So it kind of, the 2+ charge on the magnesium kind of stabilizes the negative charge there so that it won't be able to repel our incoming nucleophile quite as much. So our glycerol swoops in and attacks this outermost phosphorus. And the bond that breaks is the one on the opposite side. So this is basically an SN2 type mechanism. So let me draw the products of this. So what we have here is this bond is going to break. The electrons from that bond will go onto that oxygen. Now this oxygen already had 2 lone pairs, now it's going to have a third. And that third lone pair gives this a full negative charge. But notice that that third lone pair could exist in a p orbital. That's parallel to the p orbitals responsible for this pi bond here. And because of that, the negative charge can spread out over those 3 atoms. And so that stabilizes or dilutes the negative charge, making this whole thing a great leaving group. At the same time, this phosphate is now going to be attached to this oxygen. So here I'll draw this oxygen on the other side with its negative charge. And these lone pairs will be a bond to that phosphate. Now, if we're numbering this from the top, this is going to be glycerol 3-phosphate. And so that's the name for this, because if you're numbering this from the top, this would be carbon number 1, carbon number 2, and carbon number 3 of glycerol. On carbon number 3 of glycerol, we have a phosphate, so this is going to be glycerol 3-phosphate. Okay, so now that we have this glycerol 3-phosphate, we can we're at a position where we can turn this into something that resembles a sugar almost, and that can enter a carbohydrate metabolism pathway. All we have to do is take this hydroxy group, that alcohol there, and turn it into a ketone. And then that would resemble a smaller sugar type thing. To do that, we're going to use a different enzyme. That enzyme is SN-glycerol 3-phosphate dehydrogenase. So ultimately, we're going to be taking a hydrogen off of this glycerol 3-phosphate. And this SN there stands for stereospecific numbering, and that just means that we're going to number it so that the phosphate is on carbon number 3 of glycerol. And you can make sure that that's the case as this molecule wiggles around. Sometimes it's because it's 3-dimensional, this can wiggle around so that sometimes this phosphate would look like it's on top. To do that, you use the stereochemistry of that middle carbon there. So if you take the stereochemistry into account, you can assure that the phosphate is on carbon 3, so SN-glycerol 3-phosphate, and the enzyme is going to take a hydrogen off that, so it's a dehydrogenase. It's really just going to act as a base here. Now, to understand how this works really and completely, we have to take a quick look at one of the cofactors for this enzyme, and that's going to be NAD, or NAD plus. So that is nicotinamide adenine dinucleotide. And what's here, as we've seen with a lot of these biological molecules, there are big parts that sort of have these common pieces. Notice here how similar that is to ATP. And here we have another kind of common piece. What's unusual here and what's going to be reactive is the nicotinamide part here at the end. So that's the part that's going to be reactive. The other thing, make sure it fits in the right enzyme pockets and has the right solubility and can be made appropriately and regulated appropriately in your cells. But as far as the reaction goes, it's going to be this part right here that's reactive. So let's zoom into that and take a look at that. Notice you have the positive charge there from the NAD+. And it might not be clear immediately, but this carbon here is actually an electrophile. It is actually slightly positive. Now, to see that, you would have to see the resonance structure for this. So here you have a pi bond between two atoms of different electronegativity. That's one of the 5 resonance patterns that you would want to know. When you have that, an alternative resonance structure would be showing the pi electrons going onto the more electronegative atom. Now, if those do that, that nitrogen would have a full lone pair. It wouldn't have a positive charge anymore, but this carbon would have lost electrons, the electrons that were in the pi bond, and so now this carbon will have a positive charge instead of the nitrogen. If you look at this molecule, though, you have a second resonance pattern. Here, you have an allylic positive charge. When you have an allylic positive charge, the pi electrons move toward the positive charge. So that you get the pi bond there and a full positive charge on this carbon. Now remember, with resonance structures, none of the individual structures are the true structure. The true structure is always only the average of them all. So although in this one resonance structure we drew, there's a full positive charge here, we had 2 other resonance structures, one where the positive charge was here and one where it was on the nitrogen. So in the true structure, you'd actually have slightly positive charges on all of these, and they all would be electrophilic centers. Just because it's this electrophilic center that's going to react in this reaction, I'm going to leave this resonance structure here with that positive charge. Okay, so an important thing to note is that with NAD+, this is going to be our electrophile in this reaction. Now a lot's going to happen at the same time here. I'm going to position this base above the glycerol. That base is ultimately going to, and let me actually draw this OH on the other side here. I know that'll look a little bit awkward, but I'm going to draw it here just so that the reaction and the role of these different reagents can become a little clearer. Okay, so I'm just redrawing that glycerol. Okay, so ultimately what we want to do is turn this bond, this alcohol here, into a ketone. And it's really just a type of elimination reaction, right? Now technically it's an oxidation. When you take a single bond to an oxygen and you turn it into a double bond, that's an oxidation. But really, oxidations, turning single bonds to double bonds, are a type of elimination. Right? Oxidations are when you turn a single bond to an oxygen to a double bond to an oxygen. But anytime you form a pi bond like like that, turning a single bond to a double bond, it's an elimination reaction. And so what we're going to need is, we're going to need this hydrogen that's also bonded here to act as a sort of leaving group. We're going to do a type of E2 reaction. Now, one of the things that positions this in place and stabilizes this oxygen throughout this whole process is a zinc molecule, and the SN-glycerol 3-phosphate dehydrogenase positions that so that it stabilizes that oxygen. Notice we're seeing a lot of the common theme in this role of these metals and these enzymes. They often position things correctly, neutralize charges. That's what we saw with the magnesium earlier. So here, there's going to be a push and a pull. The pull is going to come from this electrophile from the NAD+. And just so that this fits a little better, I'm going to make it a little smaller, just so it gets closer to our molecule of interest. So the pull is going to come from that NAD+, and actually, let me just do this. It's going to come from the electrophile. This is pulling electrons towards it, and the push is going to come from these electrons here. And ultimately, it's going to push this hydrogen off the molecule. Hydrogen is usually a terrible leaving group, but here it's being manipulated by the electrons around it. So the SN-glycerol 3-phosphate dehydrogenase enzyme uses one of its amino acids as a base to steal the hydrogen from this hydroxyl group. Now, these electrons are going to move around and try to form a pi bond. That's going to give us our ketone. And the instability around this oxygen, as these electrons swirl around it, is going to be stabilized by the zinc ion here. At the same time, this hydride, that's an H with 2 electrons, is going to get kicked off as a leaving group, because as it feels the push of these incoming electrons, it's also feeling the pull of this perfectly positioned NAD+. And so these 2 electrons get pushed onto that NAD+ molecule. So that what we're left with, now it's a lot happening all at once, but what we're left with is this hydrogen no longer being on what was the glycerol, and instead being on what has become NADH. And that H is this H that was just added. And the hydrogen on the glycerol now being attached to the base, and these electrons here forming a pi bond to that middle carbon. Now I'm going to redraw that in a way that's a little bit more customary. So normally when you see this, just by convention, the bonds should be going off where they're pointing. So here you have a point in this direction, so that's where the bonds should be coming off. So there we have that carbonyl here. So this carbonyl, I'm just drawing it so it's over there. Okay, now this is no longer glycerol 3-phosphate. This is a molecule called dihydroxy acetone phosphate. Acetone is the smallest possible ketone, so 3-carbon ketone, and you can see that here, right? Acetone. If you had 2 OHs coming off of it, it would be right here and here, so dihydroxy, but on one of the OHs you have the phosphate. So dihydroxy acetone phosphate there. That dihydroxy acetone phosphate is now able to enter a carbohydrate metabolic pathway that we'll explore in a future video. But for now, what we've accomplished is we've turned glycerol into dihydroxyacetone phosphate. And that dihydroxyacetone phosphate allows it to get metabolized in your liver, broken down in your liver through a carbohydrate metabolic pathway. So here we're trying to show what happens from the time you eat a triglyceride to the time you get energy from it. This glycerol, so far we've shown what happens, how it gets through your intestinal wall into your duodenum, how it gets broken down once it gets to your cells in the form of a chylomicron, using the lipoprotein lipase, which is the same as the pancreatic lipase. And now in this video, we've shown how that glycerol gets changed into something that can be broken down like a carbohydrate. And in the next video, we'll explore how the 3 fatty acids get broken down in the mitochondria of the cell.